Part 5, Failure Patterns in Modern Systems. Chapter 17, Industrial Lipids and Structural Breakdown. Industrial lipids enter the system as structurally altered inputs that do not align with the geometric and functional requirements of biological membranes. These molecules are often modified through processes that change the position, configuration, or stability of double bonds, creating fatty acid structures that differ from those naturally integrated into phospholipids. When incorporated into the bilayer, these altered geometries disrupt the packing patterns that membranes depend on to maintain cohesion, permeability control, and organized signaling. The primary issue begins at the level of alignment. Membrane stability depends on predictable interactions between neighboring lipid chains. Industrial lipids introduce irregular shapes that do not conform to these interactions. Some configurations force chains into positions that create spacing inconsistent with surrounding lipids, while others produce rigid segments that fail to integrate into the dynamic movement of the bilayer. This mismatch alters local packing density, creating regions with inconsistent mechanical and electrical properties. These disruptions affect how the membrane behaves under normal conditions. Increased irregularity leads to uneven permeability, where some regions allow greater ion movement than others. This variation distorts electrical gradients and introduces variability into signaling processes that depend on consistent membrane properties. The bilayer loses uniformity, and the system begins to operate with localized differences in behavior rather than a coordinated whole. Protein function is directly influenced by these changes. Embedded proteins rely on a stable and predictable lipid environment to maintain their structure and transition between functional states. When the surrounding membrane contains irregular lipids, the mechanical context shifts. This alters how proteins fold, move, and interact with other components, reducing the precision of signaling and transport processes. The effect is not limited to a single pathway. It extends across systems that depend on membrane integrity. Chemical stability is also compromised. Certain industrial lipid configurations are more susceptible to modification under physiological conditions, while others resist normal metabolic processing. This creates a condition where altered lipids either degrade into disruptive byproducts or persist within the membrane longer than intended. Both outcomes interfere with remodeling cycles that rely on the ability to remove and replace components efficiently. Cholesterol interaction becomes less effective in this environment. Its ability to regulate fluidity and stabilize packing depends on the compatibility of surrounding lipids. When irregular structures dominate, cholesterol cannot position itself in a way that restores order. The membrane remains disorganized, and the regulatory capacity of cholesterol is reduced. Mechanical stress amplifies these issues. Under deformation, regions containing incompatible lipids respond differently from surrounding areas, concentrating strain and increasing the likelihood of structural defects. These defects compromise the membrane's role as a boundary, allowing uncontrolled exchange and altering the conditions required for proper function. Energy systems are indirectly affected through increased inefficiency. Variability in permeability leads to greater leakage of ions and protons, increasing the energy required to maintain gradients. This places additional demand on mitochondrial output, reducing overall efficiency and limiting the system's ability to support other processes. Remodeling cycles attempt to correct these disruptions by removing incompatible lipids and replacing them with suitable components. The effectiveness of this process depends on turnover rate and the availability of appropriate substrates. If industrial lipids continue to be supplied, they are reintroduced into the membrane, preventing restoration of structural consistency. Industrial lipids therefore contribute to structural breakdown by introducing components that do not integrate with the membrane's architecture. Their presence disrupts packing, alters permeability, and reduces the precision of signaling and transport. These effects accumulate over time, shifting the membrane away from a controlled and uniform state toward one characterized by variability and inefficiency because the structural inputs no longer match the requirements of the system they are attempting to support. Seed oils and high-PUFA exposure. Seed oils deliver a concentrated load of polyunsaturated fatty acids that shifts membrane composition through repeated incorporation cycles. These inputs do not act at the level of immediate signaling. They enter circulation, are packaged into transport systems, and are gradually inserted into phospholipids as existing membrane components are replaced. Over time, this process increases the proportion of highly unsaturated chains within the bilayer, altering how lipids pack, how they move, and how they respond to stress. The effect is cumulative. Each remodeling cycle adds a small increment, and the accumulation of these increments defines the structural state of the membrane. High-PUFA exposure changes packing density first. Multiple double bonds prevent close alignment between neighboring lipids, increasing spacing and reducing cohesive interactions. As these lipids accumulate, the membrane shifts toward a more permissive state with greater lateral movement and higher baseline permeability. This alters the containment properties of the bilayer, allowing ions and small molecules to pass more easily and increasing the energy required to maintain gradients. The system compensates by increasing transport activity, which raises metabolic demand without improving structural stability. Chemical reactivity increases alongside these structural changes. Each additional double bond introduces a site that can undergo oxidation. As PUFA content rises, the number of potential initiation points for oxidative reactions increases, and the likelihood of propagation across the membrane expands. This creates conditions where localized chemical events can spread more easily, altering multiple regions of the bilayer and generating byproducts that further disrupt packing. The membrane becomes both more reactive and less capable of containing that reactivity. Signal behavior shifts within this environment. Increased fluidity allows receptors and signaling molecules to encounter one another more frequently, lowering the threshold for activation. At the same time, reduced structural constraint weakens the boundaries that normally limit where these interactions occur. Signals become more diffuse, spreading beyond localized domains and persisting longer than intended. This introduces variability into signaling outcomes, as the membrane no longer enforces consistent conditions across its surface. Cholesterol attempts to modulate these changes by inserting between lipid molecules and reducing excessive movement. However, its stabilizing capacity depends on the surrounding structure. When PUFA content dominates, the degree of disorder limits how effectively cholesterol can restore cohesion. The membrane remains more fluid and less controlled, even with regulatory input, because the underlying composition does not support tight packing. Mechanical stability declines under these conditions. A membrane with reduced packing density cannot distribute force evenly, leading to localized strain during deformation. These regions are more prone to thinning and transient defects, increasing permeability and disrupting controlled exchange. This affects tissues exposed to continuous mechanical stress, where structural integrity is required to maintain function. Remodeling cycles determine the persistence of these changes. As long as high-PUFA inputs continue, new lipids are incorporated at a rate that sustains or increases their proportion within the membrane. Removal of these components requires time and active turnover, meaning that the structural effects persist beyond the period of exposure. The membrane reflects long-term input rather than short-term variation. High-PUFA exposure therefore alters membrane behavior through gradual but sustained changes in composition. Increased spacing, higher reactivity, and reduced structural constraint shift the bilayer toward a state that prioritizes movement over containment. This affects permeability, signaling precision, and energy efficiency because the structural conditions required for controlled function are progressively replaced with those that allow greater variability and less resistance to disruption. Oxidized fats and membrane damage. Oxidized fats enter or form within the membrane as structurally alter molecules that no longer behave like intact lipids. Oxidation changes the chemical configuration of fatty acid chains, introducing reactive groups, breaking segments, or creating cross-linked structures that disrupt how these molecules fit within the bilayer. When these altered lipids are present, they do not align with neighboring chains in a predictable way. This immediately affects packing density, increasing spacing in some regions while creating rigid, misaligned segments in others. The membrane becomes uneven at the molecular level, and this unevenness translates into functional instability. Insertion of oxidized lipids alters permeability first. The bilayer depends on consistent alignment to limit the movement of ions and small molecules. When oxidized components create irregular gaps or distort surrounding lipids, these pathways open more easily. Ion gradients begin to dissipate as leakage increases, forcing the cell to expend additional energy to restore separation. This shift does not remain localized if oxidized lipids accumulate. It spreads across the membrane as more regions adopt similar irregular structure. Propagation of damage follows through interaction with neighboring lipids. Oxidized molecules can initiate further reactions, altering adjacent fatty acids and extending structural disruption. This creates clusters of damaged lipids that behave differently from intact regions, increasing variability across the membrane. These clusters act as points of weakness where mechanical stress is concentrated and where permeability is elevated, further compromising the boundary function of the bilayer. Protein behavior changes within this altered environment. Membrane proteins depend on a stable lipid matrix to maintain their structure and transition between functional states. When the surrounding lipids are irregular or chemically modified, the mechanical support they provide becomes inconsistent. This affects how proteins open, close, and interact with signaling partners, reducing the precision of processes that rely on coordinated activity. Oxidized fats also generate secondary products that influence signaling pathways. These molecules can act as indicators of damage, triggering responses that alter cellular behavior. While this signaling can initiate repair processes, it also increases the overall activity within the system, adding to the structural and metabolic load. The membrane becomes both the source of damage and a participant in the response to that damage. Removal and replacement are required to restore structure. Enzymatic systems identify oxidized lipids and extract them from the membrane, directing them toward processing pathways. New lipids are then synthesized and inserted to replace the damaged components. This process depends on sufficient energy and the availability of suitable substrates. If removal is slower than accumulation, oxidized lipids persist, extending their impact on membrane behavior. Cholesterol and surrounding lipids cannot fully compensate for these disruptions. While they may stabilize adjacent regions, they cannot restore the original structure of altered molecules. The membrane remains heterogeneous, with areas that differ in permeability and mechanical properties. Oxidized fats therefore act as structural defects within the bilayer. They disrupt packing, increase permeability, and alter protein function, creating conditions where the membrane can no longer enforce consistent boundaries. As these defects accumulate, the system operates with increasing variability and reduced efficiency because the physical framework that supports controlled exchange, signaling, and energy maintenance has been compromised at the molecular level. Repeated remodeling into unstable structures. Remodeling is intended to preserve membrane function by removing altered components and inserting new ones that restore structural integrity. When the input pool itself is unstable, this process no longer corrects the system. It reinforces the same instability with each cycle. Fatty acids are continuously exchanged within phospholipids, but if the available substrates are dominated by highly unsaturated or chemically modified lipids, every round of replacement shifts the membrane further toward a state of reduced packing density and increased reactivity. The process that normally maintains order becomes a mechanism that embeds disorder into the architecture. Each remodeling cycle replaces only a fraction of the membrane at a time, but the cumulative effect is progressive. Newly inserted lipids alter local packing, which changes how adjacent lipids interact and how enzymes access the bilayer. This altered environment then influences the next cycle of replacement, favoring further incorporation of similar structures. Over time, the membrane transitions through intermediate states, each with slightly reduced cohesion and increased variability. There is no single point where instability begins. It emerges from the accumulation of small structural changes that are not corrected between cycles. Instability compounds because newly incorporated lipids are more susceptible to modification. As these lipids undergo oxidation or mechanical distortion, they generate byproducts that interfere with surrounding structure and signaling. Remodeling removes some of these altered components, but if the replacement lipids share the same vulnerabilities, the system cannot resolve the underlying issue. The membrane remains in a continuous loop of incorporation, damage, and partial repair without returning to a stable baseline. This cycle affects how force and energy are managed. Reduced packing density increases permeability, allowing ions and protons to cross the membrane more easily. This increases the energy required to maintain gradients, placing additional demand on mitochondrial function. At the same time, mechanical stress is no longer distributed evenly across the bilayer. Regions with higher instability absorb more strain, leading to localized defects that further disrupt function. These defects alter both structural and electrical properties, amplifying variability across the membrane. Signaling systems adapt to this changing environment. Increased fluidity and irregular packing alter how receptors cluster and how signals propagate. Activation thresholds shift and termination becomes less precise. The system begins to operate with altered sensitivity and timing, not because signaling pathways have changed, but because the structural conditions that govern their behavior have drifted. Each remodeling cycle reinforces these conditions, embedding them more deeply into the membrane. Cholesterol and remaining saturated lipids attempt to stabilize the structure, but their influence diminishes as the proportion of unstable lipids increases. Their ability to maintain order depends on sufficient surrounding alignment. As that alignment is lost, their regulatory effect becomes localized rather than global, leaving large regions of the membrane in a less controlled state. Recovery requires a shift in both input and turnover. The system must not only remove unstable components, but also replace them with lipids that restore packing density and reduce susceptibility to modification. This process takes time because each cycle replaces only a portion of the membrane. Until the input pool changes, remodeling continues to reinforce instability rather than resolve it. Repeated remodeling into unstable structures, therefore, represents a failure of corrective processes under conditions where the available substrates do not support stability. The membrane is continuously rebuilt, but each reconstruction incorporates the same structural weaknesses, preventing the system from reestablishing a consistent and controlled environment because the inputs driving reconstruction do not match the requirements of the architecture they are meant to maintain. System-wide signaling distortion. When membrane structure drifts away from controlled packing and organized fluidity, signaling distortion does not remain localized. It propagates across systems because the same structural principles govern receptors, channels, and transporters in every tissue. The membrane sets the conditions under which signals are initiated, amplified, and terminated. When those conditions become inconsistent, the entire signaling network begins to operate with altered thresholds, timing, and intensity. Distortion begins at the level of receptor interaction. Increased fluidity and irregular packing allow receptors to encounter one another more frequently, lowering the threshold required for activation. Signals initiate with less input and often without clear external triggers. At the same time, the lack of structural constraint allows these interactions to occur outside defined domains, spreading activation across larger areas of the membrane. What should remain a localized event becomes diffuse, reducing specificity and increasing background activity. Propagation of signals changes as well. In a structured membrane, signaling complexes form within confined regions and transmit information through defined pathways. When structure is compromised, these complexes form and dissolve unpredictably. Signals can be amplified in one region and dissipated in another, depending on local membrane composition. This variability disrupts coordination between pathways, leading to outputs that are inconsistent even under similar conditions. Termination becomes impaired. Controlled fluidity allows signaling components to separate once a response is complete. When the membrane is too fluid or irregular, these components remain associated longer than intended, prolonging activation. This extends signaling beyond its functional window, increasing the likelihood of overlapping or competing signals. The system loses temporal precision, and responses persist in the absence of appropriate input. Electrical signaling is directly affected by these changes. Ion channels rely on stable membrane properties to open and close in response to specific stimuli. Increased permeability and structural variability alter the gradients that drive ion movement and change how channels transition between states. This introduces noise into electrical signals, affecting the timing and coordination of activity in excitable tissues. Cross-system effects emerge because signaling pathways are interconnected. Changes in membrane structure in one tissue influence how signals are generated and interpreted in another. Hormonal signaling, immune responses, and metabolic regulation all depend on consistent membrane behavior. When distortion occurs, these systems receive and transmit signals that no longer reflect actual conditions, leading to mismatched responses across the body. Feedback mechanisms amplify this distortion. Altered signaling can increase the production of reactive species, further modifying membrane lipids and reinforcing structural instability. This creates a loop where structural disruption leads to signaling changes, and signaling changes promote additional structural disruption. The system moves further away from controlled operation with each cycle. Energy demand increases as the system attempts to compensate. Maintaining gradients, correcting signaling errors, and repairing structural damage all require additional ADP. This diverts resources from other processes and reduces overall efficiency. The system operates under increased load while producing less precise outputs. System-wide signaling distortion therefore arises from a common structural shift that affects every membrane-dependent process. As packing becomes irregular and fluidity exceeds controlled limits, the conditions that enforce specificity and timing are lost. Signals initiate too easily, spread too broadly, and persist too long. The network remains active, but its outputs no longer correspond accurately to input because the structural framework that governs interaction and containment has been altered across the entire system.